The goal of this procedure is to analyze exosomes by their size and to quantify them by using a nanoparticle tracking analysis or NTA instrument through a semi-automated method. This is accomplished by using differential centrifugation steps to obtain an exosome pellet from human blood. The exosomes are resuspended at a concentration that is predetermined for the amount of starting plasma to obtain the appropriate scatter intensity during NTA. The suspension is then filtered to separate exosomes from larger particles. Next, a calibration of the NTA instrument is performed using a control suspension containing 200 nanometer-sized polystyrene particles. The exosome solution is then injected into the NTA instrument, and the ideal setting of the NTA is selected by performing a pre-test before performing the final test. Ultimately, results can be obtained that show quantification of all measured particles and listing of the particles by their sizes.
The demonstration of this method is critical, as the dilution and the setting for each prepared exosome solution has to be performed individually to gain the best results. The measured exosomes should be in an ideal concentration, so that the live view mode displays around 600 particles per screen. Also, a pretest should be performed to find the optimal sensitivity range for the measurement. A high setting of sensitivity leads to visualization of more small particles, but also increases issues-related background noises. The procedure will be demonstrated by Anatol Maier, a grad student from our laboratory.
To begin exosome preparation, collect whole blood in 3 citrate tubes via